In these next few foundations videos, we're going to switch gears and review some basic cell biology. Now, I'm sure you're familiar with a lot of this already, but you may not have thought about it much lately, so we're going to move through a lot of this pretty quickly just to jog your memory and to focus on the cell bio topics that have a tendency to show up on step one. The first topic we're going to review are the various cell organelles, starting with the nucleus. Now, the nucleus is contained by a nuclear envelope, and it's a very tightly regulated structure. The nuclear envelope is kind of like a bouncer that guards the nucleus and keeps everybody out, because not everyone gets to party with the DNA. In fact, so the nuclear envelope is like a bouncer at a club guarding the door. He keeps the DNA inside the nucleus, but he also has to let the mRNA that's transcribed out of the nucleus. So what determines what can enter and leave the nuclear envelope? Well, the nuclear envelope has nuclear pores, and the only way to get through the nuclear pores is by having something called a nuclear localization signal. These are definitely worth knowing for your test, so let's take a look in the study guide. The nuclear localization signal is a specific sequence of amino acids consisting of 4 to 8 amino acids. It's rich in the amino acids proline, arginine, and lysine because these are more positively charged amino acids, which allows them to bind more readily to the negatively charged DNA. These nuclear localization signals are present on proteins that are headed for the nucleus, like histones, because the proteins are synthesized in the cytoplasm and then transported to the nucleus. The nuclear pores recognize these signals and allow the proteins to enter the nucleus. So if the nuclear envelope is like a bouncer that regulates who gets into the nucleus, the nuclear localization signals are like a password to get those nucleus-bound proteins past the bouncer. So last year, a student suggested that to get into the club, you have to be pals with the bouncer. And PAL stands for proline, arginine, and lysine, which are the amino acids found in the nuclear localization signal password. And another student suggested that the bouncer only lets people who bring positive energy into the club, because again, those are positively charged amino acids. Then one last point. If you have a mutation in a single amino acid in this 4 to 8 amino acid sequence, it could potentially prevent that protein from being transported into the nucleus. While we're talking about the nucleus and DNA, let's talk about the different phases of the cell cycle. Right after a cell has completed mitosis, it goes into interphase. And the first part of interphase is called the G1 phase, for growth phase 1, or gap phase 1. So cells that are designed to divide very rapidly, like bone marrow stem cells, have a fairly short G1 phase because they want to get to S phase quickly, because S phase is where the DNA is replicated. So S is for synthesis of DNA. Then once a cell gets to S phase, it's committed to cell division. But some cells are not meant to divide, so they actually go into a G0 phase. And then cells like hepatocytes and lymphocytes are on standby, and they can go from G0 to G1 in order to grow and prepare for mitosis if needed. Then after the G1 growth phase, there's S phase, and then there's a second growth phase called G2, and then an M phase where mitosis takes place. But our bodies need to be able to control and regulate the cell cycle to keep cells from just growing and dividing out of control. So there are checkpoints along the way that control the transitions between these different phases. Now, these transitions are regulated by 3 things mainly: tumor suppressor proteins, cyclins, and cyclin-dependent kinases. So let's start with a few tumor suppressors. Going from G1 to S phase, there's the RB gene and the P53 tumor suppressor gene. So both of these genes encode protein products that are critical for regulating the transition from G1 to S phase. If these proteins don't give their stamp of approval, then the cell isn't allowed to go into S phase. Now, mutations in these genes result in unregulated growth and cell replication, which is why these are called tumor suppressors. As long as these genes and their protein products are functional, they suppress the unregulated growth that we see in cancer. Now, there are 2 main cancers associated with mutations in the RB gene. We'll talk about this in detail in the oncology section, but one cancer associated with this is retinoblastoma, which is why this is called RB. The other cancer is osteosarcoma. And then P53 is also involved in regulating the transition from G2 to M phase. Next, let's talk about cyclins and cyclin-dependent kinases, or CDKs. A kinase is an enzyme that uses ATP to phosphorylate things, so to add phosphate to things. Kinases are expressed constantly. They're always around, but they are inactive unless they need to be active. Now, cyclins are regulatory proteins that control cell cycle events. They activate those cyclin-dependent kinases. Now, in order for the cell cycle to progress, these CDK and cyclin complexes have to be activated and then inactivated. Let's look at the study guide and dig into the details about how these work. Cyclins and CDKs phosphorylate target proteins to drive the cell cycle. All cyclins are degraded by ubiquitin protein ligase when their cell cycle specific job is complete. Ubiquitin is a protein that gets tagged onto other proteins to mark them for destruction. If a protein gets tagged with ubiquitin, that's a signal for this ubiquitin protein ligase to break down that protein. So because of this, ubiquitin is a very important molecule in regulating the cell cycle. You need ubiquitin in order to shut down these cyclins. Next, tumor suppressors P21, P27, and P57 bind to these cyclin CDK complexes and inactivate them. We mentioned P53 before, and P53 controls the transcription of P21. So tumor suppressors inactivate cyclin complexes and prevent them from continuing to phosphorylate things. Again, because these kinases are always around, we need to be able to activate and inactivate them at precisely the right times. So going from the G1 to the S phase, there are a few specific molecules that are worth knowing. You have cyclin D that binds and activates CDK4. Then CDK4 phosphorylates the RB protein, which allows that RB protein to be released from a transcription factor called E2F. Now with E2F unbound, the cell's free to transcribe and synthesize components needed for progression through the S phase. So those components are things like DNA polymerase, thymidine kinase, dihydrofolate reductase, and cyclin E, all of which are needed in order to synthesize DNA. And you can't get these components unless E2F comes unbound from RB, which only happens when cyclin D and CDK4 are around. Now this is a pathway you need to know for your test. It's a picky little detail, but you need to know it. Then the next bullet says that cyclin E binds and activates CDK2. And when this happens, the cell is allowed to progress into S phase. So we see two regulatory components that allow the cell to progress from G1 into S phase. CDK4 coupling with cyclin D and cyclin E coupling with CDK2. So make sure you pair those up in your mind. One student suggested that you can remember CDK4 goes with cyclin D because D is the fourth letter of the alphabet. But however you do it, make sure you remember those. Then going from the G2 phase into M phase, cyclin A and CDK2 allow the cell to progress into prophase of mitosis. And then the cyclin B and CDK1 complex is activated by CDC25, which causes the breakdown of the nuclear lamins and the initiation of mitosis. Nuclear lamins are basically the skeletal framework of the nucleus. It's a type of intermediate filament, and the skeletal framework has to be broken down in order for the nucleus to divide and for the cell to divide. That's one process that regulates mitosis. The cell nucleus holds the DNA that codes for the proteins that cells produce, but it's the proteins that actually do all the work. So let's talk about the cellular machinery that cells use to synthesize and process these proteins. We're going to be looking at some electron micrographs because you need to be able to recognize these various organelles. You're very likely to have a question asking you to identify a particular organelle. It's probably a 4-star topic, which means nearly everyone can expect at least one question on this. So let's start with the rough endoplasmic reticulum, or the rough ER. The rough endoplasmic reticulum is rough because it has ribosomes. Now the ribosomes are translating a lot of protein. Specifically, this is where we synthesize the proteins that will be secreted outside the cell, and the place where end-linked oligosaccharides are added to the proteins. So it makes a lot of sense that cells that export a lot of proteins, like plasma cells, have a lot of rough endoplasmic reticulum. So where in the cell do we make the proteins that are going to remain in the cell? Well, there are also free ribosomes suspended in the cytoplasm that are not attached to the rough ER. And these free ribosomes are the site of synthesis of cytosolic proteins and the proteins that become part of the organelles. Now, in the neurons, the rough endoplasmic reticulum is called Nissl bodies, or Nissl substance. And the Nissl bodies synthesize enzymes and peptide neurotransmitters. Now, these Nissl bodies are found in the dendrites, but not in axons. The axon hillock sometimes looks very similar to a dendrite up close. So if you look at the cell body and you see Nissl bodies, you know you're looking at a dendrite rather than an axon. And then in addition to rough endoplasmic reticulum, there's also smooth endoplasmic reticulum. It's like the Justin Timberlake of cell biology. Smooth. Smooth endoplasmic reticulum is where steroids are synthesized and where drugs and poisons are detoxified. So where would you expect to see a lot of smooth endoplasmic reticulum? In the adrenal cortex and the hepatocytes. So the adrenal cortex cells are rich in smooth ER because they're making steroid hormones. And hepatocytes are important for metabolizing and detoxifying things. So if you take a lot of medications, your hepatocytes are going to have a lot of smooth endoplasmic reticulum in order to metabolize all those drugs. So going back to the synthesis of proteins, once we've translated the amino acid sequence into rough ER, we need to make a bunch of post-translational modifications to that peptide. And those modifications take place in the Golgi apparatus by and large. But how do we get the new polypeptides over to the Golgi? You don't just turn them loose and cross your fingers and hope they find their way to the right place. Instead, our cells use something called trafficking proteins, which help the endoplasmic reticulum form vesicles that can carry the proteins to the Golgi. So sometimes you'll see these called vesicular trafficking proteins. So there are three basic trafficking proteins that I want you to know. The first two are called coat proteins because they coat the surface of the endoplasmic reticulum, or the surface of the Golgi. And then when it's time to form a vesicle, the coat proteins form spherical polymers that sort of help shape vesicles into little spheres so that you can move the proteins around inside the cell within those vesicles. So there's coat protein 2, or COP2, which is involved in the anterograde trafficking, moving protein products and steroid products forward from the ER to the Golgi apparatus. And then the COP1 proteins are involved with retrograde trafficking of vesicles, where stuff is moving from the Golgi back to the ER. So usually proteins go from the ER to the Golgi and then on outside the cell. But sometimes you need to ship something backwards from the Golgi back to the ER. And if you want to go backwards, you need a COP1 protein. And then the third trafficking protein is something called clathrin, which coats the far side of the Golgi, the trans side of the Golgi. And it helps it form vesicles to transport enzymes called hydrolases to the lysosomes. The lysosomes are little vacuoles full of enzymes that break down cellular debris and waste products. So lysosomal hydrolase actually do all the breaking down, but they're synthesized inside the rough endoplasmic reticulum, and then they're transported to the Golgi for modification, and then once they're all ready to go, the clathrin helps the Golgi form these little vesicles to move the hydrolases out to the lysosomes. Then the other big job of clathrin is to help the plasma membrane bring stuff from outside the cell into the cell. But we're going to talk about that in Foundations 5 when we talk about the plasma membrane. So we've made our proteins in the rough endoplasmic reticulum. We used COP2 trafficking proteins to move it over to the Golgi apparatus. The Golgi apparatus does a lot of modification and packaging of proteins and lipids and other secretory products to get them ready for export. Now the functions of the Golgi apparatus can be divided into two big categories. The first category is the distribution of proteins and lipids from the endoplasmic reticulum to the plasma membrane, and also the lysosomes and the secretory vesicles. So the Golgi apparatus is kind of like the cellular post office. It makes sure that everything gets sent to the right place. In order for a protein to get to the lysosome or to get packaged in a secretory vesicle, it has to go through the Golgi. The second big job of the Golgi apparatus is post-translational modification of proteins and proteoglycans, which are just proteins with saccharides or sugars tacked on. Proteoglycans are an important part of connective tissue. Now there are lots of different types of post-translational modifications going on here. The Golgi glycosylates core proteins to form proteoglycans, tacking things like chondroitin sulfate and heparin sulfate onto the core proteins. The Golgi also adds sulfonyl groups to sugars in proteoglycans and to selected tyrosine residues in proteins, and this is called sulfation. It can add O-oligosaccharides to serine and threonine residues. And then the last modification that I really want you to know is that the Golgi is responsible for adding mannose-6-phosphate to asparagine residues located on specific lysosomal enzymes. This mannose-6-phosphate is like the address label that tells the cell that those enzymes need to go to the lysosome. Again, lysosomes are where cells break down waste products and cellular debris. So the Golgi apparatus tags these enzymes with mannose-6-phosphate, which tells the rest of the cell that this is an enzyme for the lysosome. And one more time, which amino acids can be modified by the Golgi? Tyrosine, serine, threonine, and asparagine. That does get tested from time to time. Now, what happens if those lysosomal enzymes are not tagged with mannose-6-phosphate? Well, this causes something called I cell disease, or inclusion cell disease, which is an inherited lysosomal storage disorder. There's a deficiency in mannose phosphorylation, and without that mannose-6-phosphate address label to target the proteins to the lysosomes, these enzymes are secreted outside the cell. Now this results in high plasma levels of lysosomal enzymes, clouded corneas, coarse facial features, and restricted joint movement. I cell disease is often fatal in early childhood. and it looks very similar to mucopolysaccharidoses, such as Hurler syndrome or Hunter syndrome. Another important part of post-translational protein modification is protein folding. Polypeptides have to undergo folding into the proper three-dimensional conformation in order to become functional proteins. And there's a type of protein called a molecular chaperone that assists in this folding process. Chaperones assist in the proper folding and transport of polypeptides across the ER, Golgi apparatus, and beyond. So chaperones are important for the proper functioning of the ER. Some chaperones are synthesized constantly and are involved in the normal intracellular protein trafficking. Other chaperones are induced by stressors such as heat. So these are called heat shock proteins, HSP70 and HSP90. And these chaperones rescue shock stress proteins from misfolding. So if the folding process is not successful, the chaperones facilitate degradation of the damaged protein. This degradation process often involves ubiquitin, which is also a heat shock protein. Ubiquitin is added to the abnormal protein, which marks it for degradation by the proteasome. And we talked about how ubiquitin marks cyclins for degradation to help regulate the cell cycle. Well, ubiquitin also marks these abnormal proteins for degradation too. Now finally, let's talk about how we get rid of proteins that we don't need anymore. This is protein degradation or proteolysis. Now, proteolysis can be accomplished by three different means. Option one is proteasomal degradation, which is where the protein is broken down by a structure called a proteasome. This process uses those ubiquitin tags again. So ubiquitin is attached to the defective proteins to mark those proteins for breakdown. Then the proteasome comes along and it contains an enzyme complex called ubiquitin protein ligase, and that recognizes the ubiquitin tag and then destroys the protein. Option two is lysosomal degradation, where the protein is sent to a lysosome and the lysosomal enzymes digest it. And we discussed that one already. And then option three for proteolysis is to use calcium-dependent enzymes that aren't found in the lysosome and aren't found in the proteasome, but again, they break down proteins. So there's a proteasomal mechanism which uses ubiquitin, there's a lysosomal mechanism, and there's a calcium-dependent enzyme mechanism. Three different ways to break down proteins that you don't need anymore. The next organelle I want to discuss is the sarcoplasmic reticulum. So this is a specialized kind of smooth endoplasmic reticulum that's found in muscle cells, both smooth muscle cells and skeletal muscle. So the sarcoplasmic reticulum stores calcium and it manipulates calcium balance in order to regulate muscle contraction. Calcium is one of the most important substances in the cell. Calcium is generally what makes things happen. Calcium allows muscle to contract. It triggers exocytosis of secretory products like neurotransmitters and hormones. So calcium balance within the cell is incredibly important, and the sarcoplasmic reticulum is an integral part of maintaining that proper calcium balance. Another incredibly important substance within the cell is ATP, which your cells need for energy. Now, ATP production takes place in the mitochondria through the TCA cycle and the electron transport chain, both of which take place within the mitochondria. So mitochondria are the powerhouses of the cell. Now, some cells are specialized in that they don't have very many mitochondria, less than 100. Other cells have more than 1,000 mitochondria. Mitochondria have an outer mitochondrial membrane, an intramembranous space, and then an inner mitochondrial membrane. And both of those membranes are lipid bilayers. Mitochondria are also interesting because they self-replicate, independent of what's going on in the nucleus, because mitochondria have their own DNA. No matter what DNA you got from your dad, you got your mitochondrial DNA from your mom, because all the mitochondria you had when you were a one-celled zygote came from your mom's ovum. So you can use mitochondrial DNA to trace groups of individuals that have a common maternal origin. And the mitochondria also play an important role in apoptosis. That'll be covered in Foundation 6. There's one other organelle I want to talk about, which is the peroxisome. Peroxisomes are involved in a lot of different metabolic processes, and I want to highlight three of those processes for you. First is the breakdown of very long-chain fatty acids through beta oxidation. Now, all the fats that you ingest are not the same. They're saturated fatty acids, monounsaturated fatty acids, polyunsaturated fatty acids, long-chain fatty acids, short-chain fatty acids, and so on. So the very long-chain fatty acids are broken down in the peroxisome by way of beta oxidation. The second important process is that the first steps in the synthesis of plasmalogens take place in the peroxisomes. Okay, great. What's a plasmalogen? Plasmalogens are a type of phospholipid that are found abundantly in myelin, in neural tissue. So I want you to bundle those terms together in your brain. Peroxisomes make plasmalogens, which are found in myelin. Then a third function of peroxisomes is that they contain oxidases and catalase, which are used to metabolize toxic substances that enter the cell, such as ethanol. So without peroxisomes in your hepatocytes, you would not be able to metabolize alcohol. So that's it for this video. Now we're ready for the end of session quiz. So pause the video, answer those quiz questions, and then we'll go through the answers. All right, first question. What must be present on a protein in order for that protein to gain entry into the nucleus? It's called a nuclear localization signal. Next, which cyclin CDK complexes assist in the progression from G1 phase to S phase? That was a complex of cyclin D plus CDK4 and also the complex of cyclin E plus CDK2. Next question, which cyclin CDK complexes assist in the progression from G2 phase to M phase? That was the complex of cyclin A plus CDK2 and the complex of cyclin B and CDK1. Next, what molecule does the Golgi apparatus add to proteins in order to direct those proteins to the lysosomes? It's mannose-6-phosphate. The last question, what are the different methods that a cell uses to break down proteins? So the three different methods we discussed for proteolysis are degradation by proteasome, which is mediated by ubiquitin, degradation by lysosomes, and then degradation by those calcium-dependent enzymes. All right, that's the end of Foundations 3. I'll see you next time.